Now that we've learned a little bit about the basic bits of atoms, how atoms bond together to make molecules, we need to look at those major molecules that are found in all living things, those 4 macromolecules. Now, when we're doing this, we're going to just briefly touch a little bit on organic chemistry. And when we're making this distinction or classifying a molecule as being organic or inorganic, the primary atom that we're looking for is carbon. And there's a good reason why carbon is used to determine or to distinguish between whether or not a substance is organic or inorganic. Carbon has an unfilled outer shell. That unfilled outer electron shell means that carbon is going to want to make lots of bonds. So we tend to find carbon in a lot of those molecules that are important to all living things. Things like carbohydrates, proteins, nucleic acids, and my personal favorite, lipids. Now, when we're looking at these macromolecules that are found in all living things, what we want to do is we want to look for the monomer, that smallest unit of the macromolecule that still has the properties of that molecule. The prefix mono means one, so I think of a monomer as being one chunk, one unit, that smallest unit that still has all of the atoms and all of the properties of that larger macromolecule. Now, because biological systems are all about making and breaking bonds, making and breaking molecules, it's important that we know how we can take a great big polymer, which is lots of little monomer units all linked together, and break them down into their smallest components. Those small monomer units will be what cells will be using in their biological processes. So when we're looking for those individual monomer units, and then looking at the larger polymers, those larger macromolecules, I like to arrange the information into this nice little chart because it helps me to keep everything straight and helps me to really understand how the smaller monomer units come together to make a polymer. And then with the examples, helps me to understand where I'm seeing these and have been using these in my own personal life. So I first want to start with carbohydrates. Now, our carbohydrates are really delicious and wonderful. The smallest unit, the monomer for a carbohydrate, will be a monosaccharide. Again, mono means one. And that ending saccharide or saccharide refers to sugar. So the term monosaccharide really literally means one sugar. For this class, the example of a monosaccharide that you have to have memorized is glucose. And that's because in chapter 6 with photosynthesis, we're going to spend the entire chapter talking about how plants are using sunlight energy, capturing it and storing it in the form of glucose. And then in chapter 7, we're going to spend that whole chapter talking about how cells will break the glucose molecule and continue stripping away and removing things from it in order to make ATP energy, and that's aerobic cellular respiration. So our chemical formula for glucose is C6, in other words, we have 6 carbons, H12, that's O6, and that's 6 oxygens, all bonded together into what to our tongues tastes like a very sweet molecule. Now, what I have here in front of us is an example of a polymer. This is sucrose. It is a disaccharide. It's 2 monosaccharides that have bonded together. Now, hopefully you're picking up on this idea that the ending sucrose, that OSE ending, glucose, OSE ending. Usually whenever we use that OSE ending, we're trying to indicate that we have a sugar of some kind. So sucrose, table sugar, is a common disaccharide in our environment. Now, if we keep bonding those monosaccharides together, we get larger and larger molecules. This is where we come across one of our first important themes in biology. And that theme is when we have a linear straight molecule, that molecule is going to be really hard to break apart. Lots of molecules can pack really tightly together, and those linear straight molecules all packed tightly together are really, really difficult and hard to break apart. And that's why I have some of these other examples here. So here we have a great big old chunk of wood, and this is primarily made of cellulose. It is a polysaccharide, and with cellulose, each individual monosaccharide has bonded together to make a linear straight line. We pack all of those straight lines together in our cell walls, and eventually, after many, many years, we end up with this solid, tangible wood structure. When you think about how important wood is, wood builds our houses, wood is everywhere around us, and it's going to take really high temperatures to break apart the polysaccharides into their individual monosaccharides. We can't actually eat this and break it into those small monomer chunks and get any energy out of it. I also have some Swiss chard. These plant cell walls are also made of cellulose fibrils, but the cellulose fibrils are a little bit lighter. They're a little bit easier for us to break apart, but let's face it, sitting down and eating a whole plate of this does not seem very delicious. It's going to take a lot of work to get the energy out of it. And even though Melga's kitty here thinks that he wants to eat this grass, there's no way that his body is going to be able to break down the polysaccharide into individual monosaccharides. It's not going to be able to break the big molecules apart and get any energy out. So a big question is, why can a cow eat a bunch of grass, a bunch of these linear cellulose molecules, and get energy out and be able to transform it into their body, while Melga's over here is just going to barf it up later, hopefully not on my pillow? And when we think about a cow's anatomy, a cow has to have a really specialized stomach. The stomach has 4 chambers. In each chamber, there's a different set of enzymes and bacteria. So when the cow takes a bite of grass, that grass then goes into one chamber of the stomach where enzymes and bacteria work on it, trying to loosen the polysaccharide into monosaccharides. They erp it up, they chew on it again, they put it into a new chamber, different bacteria, different enzymes working to release and loosen the bonds between each individual monomer. They do this over and over and over again. It takes a lot of time and a lot of effort to get the energy back out. So, great way to store a bunch of energy if you're a plant, but when it comes to consuming that plant and transforming it into the energy needed to run your biological processes, that's just going to take a lot more effort. This is why we love those molecules, those polysaccharides that have a lot of branches in them. The polysaccharides like starch on the potatoes. It's so easy to get in and snap each individual piece off. And when we snap those monomer units off, we can boil those right into our cells, do aerobic cellular respiration, and get lots of energy. So on our chart, what I did was I added our polymers, our disaccharide, and you could add to that sucrose or table sugar. We have cellulose and starch, some of those larger polymers, where all of our individual monosaccharides have been bonded together. Another big category of macromolecules that we find in all living things will be proteins. Now, the smallest unit of a protein is an amino acid monomer. There are 20 different types of amino acid monomers. When we get amino acid monomers bonded together, we're going to have a polypeptide. That prefix poly means many. And so polypeptide is just telling us that we've got lots of individual amino acid monomers bonded into a large molecule. I want to stop there with our proteins because in chapter 11, we're going to talk about those 20 different types of amino acid monomers and how they come together to make a protein. Another big category will be our nucleic acids. Now, the smallest unit for a nucleic acid is a nucleotide. If we have the polymer DNA, our nucleotide monomer, we have 4 different types. Adenine, thymine, guanine, and cytosine. If we have the polymer RNA, our 4 different types of nucleotide monomers will be adenine, guanine, and cytosine, but the big difference is that we'll have uracil. Now again, with chapter 11, we're going to talk a lot about nucleic acids, how they come together in order to make DNA and RNA, and then ultimately get back to that way that the DNA and RNA allows the cell to be able to make a protein. Now our final category that I wanted to end with today are the lipids. And I know that in our common language, when we start to talk about human diet or even diet for these little critters here, that one of the things we tend to do is we tend to have some baggage associated with lipids. Lipids have a lot of energy packed into the bonds between individual atoms. If we get too much excess energy in our body's going to do the smart thing. It's going to store that energy in the form of lipids, because it's as if our body is anticipating, wait a minute, there may be a day in the future where I don't have enough energy to run all the processes inside of my cell. I may need to get one of those lipids and start breaking it apart and use that energy. So inside of our bodies, lipids are a wonderful, wonderful storage molecule. But a lot of us have a few more lipids than what health officials would like us to have, and so there's some baggage associated with the lipids. I don't want you to see these as bad molecules. They're actually really, really, really amazing. And without the lipids, we wouldn't have a cell. And without a cell, we don't have life, because remember, the cell is the smallest unit of life. So, our lipids are a little strange. For our lipids, we have two monomers that have to come together. The first monomer is a glycerol group. Now, our glycerol group is not something that I'm going to talk much about in this class, but it is important if you get into the process of soap making. The other monomer that has to come together to make a lipid is something called a fatty acid chain. Now, a fatty acid chain is just lots of carbon atoms bonded to hydrogen atoms. But that fatty acid chain is going to determine how we classify our lipid, and more importantly, how that lipid is going to behave. So one lipid polymer is something called a saturated lipid, or a saturated fat, or a saturated oil. And essentially what we're referring to is that fatty acid chain. With a saturated lipid, or fat, or oil, every carbon is saturated with hydrogen. Every carbon has bonded to as many hydrogen as possible. What this does to the shape of the fatty acid chain is it makes that shape linear and straight. We get lots of linear and straight molecules all together. They can pack really, really tightly together. When molecules pack tightly together, it's really hard to break them apart. So I have some examples of saturated fats here just from my own kitchen. The first one is some margarine, and then we have coconut oil and palm oil. Now this palm oil, I don't know if you can see from the video, this is just a solid chunk of lipid inside of this container. It's not moving anywhere. And it's the same thing with this coconut oil. This coconut oil is just a solid chunk inside here at room temperature. It's not going to move anywhere. Every carbon in the fatty acid chains are stuffed with hydrogen. It's forcing those fatty acid chains to be straight. They're packed really tightly together. They're really hard to break apart. Now, when my body goes to try and eat and consume these saturated fatty acid chains, it's going to have to treat them differently because of that linear straight shape. But if I'm baking and cooking, I love that linear straight fatty acid chain. That's what allows me to make biscuits feel really crumbly. It's what allows my homemade pasta to come together and stick together in that wonderful, delightful way. And then when I eat it, it just melts across my tongue, because of course my tongue is much, much, much hotter than room temperature. And that hot body temperature is going to change and start to manipulate the shape of those fatty acid chains. So all three of these are considered less healthy. In addition to butter, all those saturated fats, our body is going to have to treat them differently. It's really not something we should be consuming a lot of on a regular basis. So the other type of fat or lipid will be an unsaturated fat or lipid. And again, what we're doing is referring to that fatty acid chain. And what happens is we have a double bond between the carbon atoms. That double bond between the carbon atoms in our fatty acid chain molecule to be kinked. And when that molecule is kinked, the molecules can be fluid and slide around one another. When we eat them, our body is going to treat them differently. So here I have some sunflower oil, and you can see as I move this around, all those unsaturated fatty acid chains just sliding back and forth. It comes down to the double bond between the carbon, causing that fatty acid chain to be kinked and allowing things to move and slide. Now, our final lipid that's going to be important is our phospholipid. Now, with our phospholipid, I'm going to draw it right under here so it's easy for you to see. We have that hydrophilic water-loving head, and we have two fatty acid chains, our hydrophobic tails. Now, one fatty acid chain is going to be saturated and straight and stable. The other fatty acid chain is going to have that double bond, that little kink. That double bond, that kink, causes that fatty acid chain to be a little bit looser, a little bit more wiggly. And so within one individual phospholipid, what we have is a lot of molecular tension. Part of it's going, ooh, where's the water? Where's the water? And the other part is going, ooh, I hate the water, hydrophobic tails. And then within the hydrophobic tails, we have one straight and stable fatty acid chain and one kinked, unsaturated fatty acid chain. So we have a second level of molecular tension within our tails, within our fatty acid chains. We get lots of phospholipids all bonded together. One on top, one below. We get a bilayer. We end up with a plasma membrane. One of those plasma membranes could be a cell membrane, essentially the outer layer of our water balloon, our cell. Phospholipids determine whether or not we have a cell. They are so incredibly crucial, which is why I wanted to end with lipids and focusing in on how incredibly important they are. Because even though we have a lot of baggage with them, as far as our diets and our personal health may be concerned, the lipids are absolutely crucial and necessary for life. If you have any questions, please make sure to email me.